So today I'll start giving you a keynote lecture on kind of the basics of phage genomics, of phage genome diversity, actually. And before I do that, I just want to tell you what is phage annotation. So we start with nothing, and then we just extract nucleic acid and we try to sequence it. And once you've sequenced it, you get this email from your sequencing facility saying, here are your sequencing reads. So the first step you need to do is look at those, do some quality control, and come to an assembly. This assembly can be in multiple formats. You can have one contig, you can have multiple contigs, but it's basically just one long stretch of DNA that you have, and then you need to come to an annotated genome. Now, this step seems straightforward, but it entails quite a couple of steps, and during these 3 days, we'll have a closer look at some of these steps. The first thing that we kind of need to do once we go from the assembly and have our contig, before we do our actual annotation, is trying to find the genome ends, because you want to have your phage genome complete and in the correct orientation before you do a lot of steps that you will have to repeat if you don't have your correct genome organization. So then we do gene prediction. So then we have a whole bunch of coding sequences. We can annotate special features like promoters or terminators and the likes, and then put it all together in this gene prediction. Oops, I clicked on something. Okay, the next step then, if you want to publish your genome, is basically formatting it in a way that we can submit it to a database. These are some of the concepts that will be used over the course of the 3 days. The first one is assembly, where you go from your reads to your contigs. Then you have read mapping, which is where you start with your contig in one file or your genome in one file, and then with your reads in another, and you come to a read mapping file or visualization, or we'll use another method today as well. And then there's the annotation, which again starts with your contig or your genome, and at the end you have this beautiful colored picture with all of your features in it. So where does the phage diversity come into play? So there's lots of different types of bacteriophages out there. And this is an important thing to keep in mind when thinking about sequencing and genomics. The first thing is that not all phages consist of DNA. There are also phages that consist of RNA. If you are lucky, you will find one of these, and they're really cool. But obviously, if you find some of these, you can't do normal sequencing and library preps because you're starting with RNA. And the two best characterized RNA phages are the double-stranded RNA phage Phi6 and the single-stranded RNA phage MS2. What is really pretty cool about Phi6 is that it is segmented. So its genome is in 3 linear pieces in the viral capsid, meaning that once you start assembling this, you will never get one contig with one genome because it's in 3 physical pieces. So that's the first thing to think about when dealing with RNA phages. Then there's the DNA phages. So I'm going to start with the ones that are not commonly found, but that doesn't mean that they're not very interesting. So probably the most sequenced phage at the moment is Phi X174 because it's used as a a spike in for Illumina sequencing, or it has been used as a spike in for Illumina sequencing a lot. So that's a very small single-stranded DNA phage with a genome of 5.3 kb. And these ones, the microviruses and the endoviruses, these are ones that are that have properly circular genomes. So the single-stranded DNA molecule is one circular molecule. This is important to keep in mind because there are certain methods, such as multiple displacement amplification, which preferentially amplify this type of genome. So if you have a DNA extract and you have, for example, a metagenome and it's not high enough quantity and you want to increase the quantity and you use multiple displacement amplifications, these type of viruses will pop out. Now, if you just do phage genomics, that's not that much of an issue. But as a side effect, if you are working with these type of phages, for example, there's M13 for E. coli, and there's the Pseudomonas phage PF1, which is a prophage, you can use this bias to actually amplify it and make sure that you have enough DNA to sequence. So that's also important to think about when we're talking about phage diversity. There's a lot of double-stranded DNA phages, and there's some cool ones that are non-tailed. So we have the corticoviridae, plasmaviridae, tectoviridae, and autoliciviridae at the moment to find. So these, some of these, like the corticoviridae, these are the only ones with circular double-stranded DNA. So that's very important to consider because a lot of people describe their genomes as being circular, whereas they're actually linear. Not this one. This is properly circular. And then we have our all-time favorites, the tailed phages. So we have multiple different morphologies of them and multiple different genome organizations. And often these genomes are displayed as a circle, as you can see for Bacillus phage SPO1, which is kind of the type phage for the Hirelviridae family, or for the genome of phage T4. These are not actually circular. In the phage particle, in the capsid, these are linear molecules. And I will keep repeating this because it is, the biology is important for me, not just the genomics. So when we sequence them, we can use sequence-based detection for phages in a whole bunch of different situations. But what we'll focus on for the sake of this workshop is the phage genome sequencing in particular, meaning that you first have a phage prep and you will make sure that that's as clean as possible, then do your DNA extraction or your RNA extraction. But for the sake of the rest of this lecture, let's just assume it's DNA, and then we will sequence that. So metagenome sequencing, prophage sequencing, single gene sequencing, they all share some features, but we will focus on the phage genome sequencing here. But I needed to have a little bit of a slide to plug the book chapter that we wrote a while ago. So for the majority of phages, the sequencing platform and the methods that you use to assemble your phage your reads, they don't matter that much. And there are a couple of things to keep in mind. For example, phage genomes are small in comparison to bacterial genomes and definitely in comparison to eukaryotic genomes. So the smallest sequencers, they're mainly sufficient for bacteriophages. You don't need 100 or 1,000-fold coverage to get a good bacteriophage genome. So what you can do is if you know that there are a lot of different phage types that you have, you can actually sequence them in the same sample without barcoding them, because they will assemble into separate genomes. And these things are well addressed in the paper by Branko Ritman and colleagues in Phage. So there's also, I mean, long-read technologies, and specifically nanopore, have become really popular. And they have certain advantages, like you can get the whole genome in one read, for example, but they're usually still more expensive. And nanopore can be computational, expensive to run on a certain laptop if you want to do your base calling at the same time and such. It's all down to how much money you have to spend and what your setup is at your lab or in the sequencing facility that you contact. One of the things that I want to point out is that Nextera library preps are quite popular, but they are transposon based. And that means that the transposon cannot insert at the very end of your genome, and the very end of your linear genome will be missed. This is not an issue when your genome is circularly permuted, and we'll get back to this later. When it comes to assembly of genomes, it is important to choose tools that are both accessible to you and in line with your bioinformatics skills. Now, this is sometimes a bit of an issue because the easier to use, the more expensive they are. So what we will show you today is all based on freely available software, but then it is more tricky to get started. Let's call it that. So one of the things that I always focus on for people is to do their read mapping, to look at their genomes, to find assembly errors, find genome ends, and we will show you a tool to do this today. So one of the things that most phages have been sequenced with is with short reads. So then you have short reads to assemble into either one contig or multiple contigs. You can also do long read assembly. So your reads are longer and you would usually use a different tool to assemble them. And then the combination of the two, where you provide your assembler with both short reads and long reads, is called hybrid assembly. So in all of these cases, if you have a very clean library prep and you have a very clean phage prep, then this may assemble into just one contig, but it could be that it's multiple contigs, and then you need to do some troubleshooting. I'm using a paper from Sutton and colleagues in microbiome from a couple of years ago, where they tested a whole bunch of assemblers. Assembly software on a virome, but what they did is they spiked their sample with Lactococcus, I think it was Lactococcus phage Q33, which you can see here at the top. And then they assembled their metagenomes and checked whether they could pull out the phage genome from their spike-in. So what they see is they tried, they showed here 6 different assemblers, and they showed that Spades and Megahit were really good at it. But what you need to remember is that an assembler will basically start randomly putting reads together, but it doesn't know strand direction, and it doesn't necessarily know where to start and where to stop. So what you can see here is that these blocks are collinear blocks of shared sequence identity. So the collinear blocks are collinear everywhere, but the sequencer will randomly start at a certain certain base. And that can be different for each sequencer. And they saw that, for example, this Ray Meta kind of inserted some random stuff, and CLC genomics was a little bit shorter, and Genius added a little bit here in comparison to the reference. And then some of them, because it's kind of a 50-50 in which direction the strand goes, so some did it in the reverse. So that, and I want to use this to show that there are assemblers that are better than others, but none are absolutely perfect. So always do some sort of manual verification of your assembly. Now, that being said, the assembler that we will use today is actually not on this list because it's relatively new. I quickly wanted to also have a word about transduction, which is the process of phages packaging host DNA and what the consequences are for genomics and sequencing and assembly. So you have your phage, it infects a host cell. You have your phage DNA, it can integrate, and it can, for example, some of those that integrate do specialized transduction where they could package a little bit of host DNA together with the phage DNA, so the joining ends of the host into the phage capsid. There's also generalized transduction, which is quite important to think. Some phages are quite efficient generalized transducers, meaning they randomly package a subset of particles, randomly packages host DNA. And then no matter how clean your phage prep is, there will always be some bacterial DNA present, which means that once you assemble, you could always have some bacterial contigs present. And you will, that doesn't mean that you've worked in a contaminated way. That's just inherent to this particular phage. But we don't know that beforehand. So I always say, don't be put off by having bacterial contigs in your assembly. So one of the things we do is mapping back reads to the phage genome to evaluate certain features of the genome. So when you do read mapping, for phages, again, the choice of read mapper is not that important, especially for the majority of phages. If you have multiple contigs, read mapping and checking the coverage is important because that is the first clue to which contig belongs to your phage, or which contigs belong to your phage, and which are bacterial contamination. The one with the highest coverage is usually your phage genome. You can also use read mapping to check for assembly errors, and it can be used to determine the genome ends, which is what we will do today. So I'm coming back to the phage genomic diversity and the phage genome structure and what the implications are for assembly. So the first, so we're now talking only about a double-stranded DNA tailed phages, and they come in multiple different genome structures. So the first one that I want to cover is the genomes that are circularly permuted. So I included one of the first genomes that I did myself for my PhD, which is DKFH limestone 1, which is circularly permuted and is packaged through headfold packaging with random ends. Another type is packaged with pack sites, where your start is fixed of the first molecule that is packaged, and you can see that in the middle. So you have each phage capsid gets packaged from your pack sequence until it's full, which is kind of more than 100% of the genome. And then in the next phage particle that's produced in the same cell, we start from the point where we left off in the previous particle, and we package more than 100% again. And then in the next phage particle, more than 100% again. So that's the principle of having a circularly permuted genome, meaning that every single particle has packaged a slightly different version of the DNA. So while it's linear, when you put it all together, it comes together as a circularly permuted genome, because each particle has a different start and stop. Or in the case of the Phage sites, they have within the same cell, they have the same start from a Phage site. So how can you see that from your read mapping? If you have headful packaging with random ends, you just have even coverage across the contig, across the genome. And with even, I say it's not 100%, there's always kind of some variation that's inherent into sequencing. If you have Phage sites, then you usually see an increased coverage immediately downstream of the Phage site, which then tapers off. And this gives you the directionality of how the genome gets packaged into the phage capsid. Then you have genomes with defined ends. And there's two main types of defined ends. There's cohesive ends, and they can either have a short 5' overhang or a short 3' overhang with a cos site on the left-hand side, or we can have genomes with terminal repeats, and they are usually direct terminal repeats, not inverted terminal repeats, where each end of the genome is flanked by an identical sequence, which when it's too long and you sequence it, it can be mistaken for a circular genome, a circularly permuted genome. So how do we see that with read mapping? When with the cohesive ends, it's usually a decreased coverage near the ends. There might be a peak at the overhang, but that's not necessarily the case. Or in the case if we have terminal repeats, that means that there's one sequence of the genome, the terminal repeats, that's present twice. Now the assembler will only display it once usually, meaning if you then map your reads back to it, there'll be twice as many reads for this region as for the other region. So you have your double coverage at the location of the direct terminal repeat. When you have defined ends, one of the things you can do to exactly determine where the defined end is using a process called primer walking or runoff Sanger sequencing. So basically you have your DNA that you've extracted, and you design primers in the location where you think the direct terminal repeat is. If you design a primer that's in the terminal repeat, then it's actually present twice, once on the left-hand side and once on the right-hand side, here and here. If you sequence, then your Sanger sequencing chromatogram will fall to 50% of the chromatogram height at the moment where your terminal repeat turns into your genome. If you design your primers outside the terminal repeat facing outwards, then your Sanger sequencing chromatogram will fall to 0. Absolutely 0, because it just runs out of genome. And this is a way where, in which you can accurately define your genome ends. And in order to call your genome complete, and when you know it's a genome with defined ends, this is something that you should actually do. So once you have all of this information, it's very important to reorganize your genome so that it's actually correct. So when you have defined ends, there's no question on where you need to have your genome start and stop. One of the things that have emerged as the consensus is that you, in your final file, have your terminal repeat, if there's terminal repeats, have it present at the front, and then indicate that this is your terminal repeat sequence, and then not have it present for the rest of the genome. If you have a circularly permuted genome, things are much more complicated because there's really no way of knowing where your genome should start and stop. So what you can do is you can compare your genome with a relative in the database. If it's well described there, you can just choose to do the same thing. So you can rearrange your genome to be collinear with the best annotated relative. Sometimes these decisions are described in literature from quite a while ago. Sometimes it's up to you to do this. And obviously what you want to do is try to find an intergenic region where it makes sense to start your genome. And some of the cyphoviruses, for example, have all been started at the terminase small subunit. So hopefully we have enough good quality genomes in the database for most of you to be able to make a good informed decision on where to start your genome. And once you've done that, only then you're ready to start with the actual annotation.